Hello, dear friends. Today we are going to discuss and study about the antibacterial. Especially, we are going to focus on to antibiotics. So, when we are going to discuss about the antibiotic, it's very important to understand what exactly the meaning of microorganisms and how they have been responsible to have an infection. So, it's a very general phenomena to discuss the microorganism versus human life, as well as to study antibiotics versus infection. In continuation to understand the definition of antibiotics, the little meaning is it's against life. As the biological concept of survival of the fittest, in which one organism destroys another to preserve itself. That's a very natural phenomenon, my dear friends. In 1942, the scientists had defined an antibiotic or antibiotic substances is a substance produced by a microorganism which has the capacity of inhibiting the growth and even destroying other microorganisms. That's a very general phenomena. You might have heard about the penicillin, the history of penicillin, the accidental discovery of penicillin, the fungi which has been developed by the antibiotic, which has been considered as an antibiotic to inhibit the growth of other microorganisms. In continuation, after definition of antibiotics, it's very important to understand what exactly the properties which has been which are very important for the antibiotics. It is a product of metabolism. It is a synthetic product produced by as a structural analog of naturally occurring antibiotic. It is analogizes the growth or survival of one or more species of the microorganism. It is effective in a low concentration. Another important aspect while studying antibiotic is chemical classification. Most of the time you will find out the classification based on the mode of action, but this classification will majorly focus on the chemical way of classification. The major class is a beta-lactam and beta-lactam is inhibitor. Another is aminoglycoside, another is a tetracycline. The next one is the macrolides, then it's a lincomycin, peptides and polypeptide, chloramphenicol, quinolones and miscellaneous. I usually suggest to follow this sequence to remember that there is a PC to ATM line queue. In Hindi if you remember that अगर आपको पैसे चाहिए तो आपको ATM के लाइन में या फिर क्यू में लगना पड़ेगा. So that's a very common logic that I used to remember the classification of these antibiotics, especially chemical classification. So what this P stands for? This P stands for the penicillin, C stands for the cephalosporin. If I can remember the two species, another P is a peptide and another C is a chloramphenicol. So there are the two species. One is a penicillin, another is cephalosporin. Another, the next P is a peptide or polypeptide, and another C is a chloramphenicol. A stands for aminoglycoside, T stands for tetracyclines, M stands for the macrolides, LIN stands for the lincomycin, and Q stands for the quinolines. So let's understand what are the general example that has been implemented into clinical practice. Amoxicillin, ampicillin, cefuroxime, clavulinates, carbapenems. This color indicate these are the subclassification of the beta lactam class. Beta lactam is a very unique class, as every class of this antibiotic is a very unique. Everyone is having its own pros and cons. So we'll study one by one. But first we'll focus on to this classification, that is a PC to ATM line Q. So moving ahead, the how exactly they work. Yes, class, subclass, and action mechanism. So these penicillin and cephalosporin, they are cell wall cross-linking inhibitors. The beta lactamase inhibitors are these are the inactivators, or they will inhibit the beta lactamase. Aminoglycosides are nothing but they are having tendency to inhibit the 30S subunits, or they I must say they bind with the beta 30S subunit, and they will inhibit the protein synthesis and fidelity. Tetracycline is another class. As the name indicates, tetracycline is contain the 4 cycle. These are also having tendency to bind with the 30S subunit, and they will inhibit the protein synthesis. These are some unique examples: tetracycline and democycline. The next important class is the macrolides. As the name indicate, macro, it means a larger group. They are having tendency to bind with the 50S subunit, and they will also inhibit the protein synthesis. The examples is erythromycin, clarithromycin, and azithromycin. You might have heard this azithromycin in this corona pandemic. The next class is lincomycin. They are also having ability to bind with the 50S subunit, and they are having tendency to inhibit the protein synthesis. The examples are lincomycin and clindamycin. This lincomycin will stand over like this, or as I have explained in the classification. The next major class is peptide and polypeptide. You might have heard about the words peptide and polypeptide when you are studying amino acids, proteins, and all these stuffs. So they are quite correlated with. But in this segment, these are playing as a role of antibiotics. So how this polypeptide will work? They will work by inhibiting the cell wall synthesis and cell membrane function. The examples are vancomycin, bacitracin, polymyxin. As the poly means has been correlated with the, like polypeptides. There is a next class which is totally independent, that is a chloramphenicol, and they are having tendency to bind with the ribosome, and they will ultimately inhibit the protein synthesis. The another independent class is the quinolones. They are having to bind with the bacterial DNA gyrase, or you can say topoisomerase. The example is the norfloxacin and ciprofloxacin. When it comes to the miscellaneous class, there are the various groups of this class, and there are some certain examples are novobiocin and the linezolid. Linezolides is a very recent trend bug antibiotic, and they are generally considered as a very higher class of antibiotic. And it is always very preferred that you should start with the lower class of antibiotic because of the resistance. Resistance is another very different and very interesting topic, my dear friends. It will need hours and hours to study about what exactly the resistance means and how they has been developed, and it's a very moral responsibility of physician as well as a pharmacist to create less chances for the resistance. So as I said, there's a very unique topic to study about the resistance, and obviously we'll require another class to study about the resistance. But for this class, you just remember the classification of the antibiotic, as we had to study about the space C2 ATM link Q. As I said previously, the previous slide was all about the chemical classification of the antibiotics. By considering the same thing, I will explain the mode of action of different antibiotics. If you consider the cell wall, before going to study this classification, you must focus on the different components of the cell. Cell wall is there, cell membrane is there, then this is DNA RNA synthesis assembly is there. We can say simply protein synthesis. You might have studied in your 10 plus 2 level or 12th level about the protein synthesis, about the 30s ribosomal unit, 50s sub ribosomal unit. You might have studied about the RNA synthesis. Then you might have studied about the DNA dependent RNA polymerase. You might have also about the DNA gyrase inhibitors and all these terms. So these are the very the basics are always there. You should keep the basics with your mind. It should be very handy to you. So whenever you can, you remember that. So let's start with the cell wall synthesis. So the very first class that we have studied into the chemical classification is the beta lactam. So they are considered as a cell wall synthesis inhibitor. Glycopeptides are there, phosphomycin is there, bacitracin is there, alpha phospholin is there. And then the next subclass, by considering the mode of action, is the cell membrane synthesis. These are the lipopeptides, and they are having ability to disrupt the cell membrane synthesis. The another class that I have not mentioned in the previous slide is the sulfonamide. They are the folate synthesis, and they are having structural dissimilar with the sulfonamides. This is another unit, my difference. So let's focus on this protein synthesis. Most of the class that are 30s and 50s subunit inhibitor, and actually the ultimate aim of this is to inhibit the protein synthesis. And as I have said, into the ATM, like aminoglycosides are there, tetracyclines are there, macrolides are there. This is a miscellaneous class. This lincolnosamides are there, amphenolicols are there, oxalidones are there. There are numerous classes, but what exactly we have to focus on to this is like ATM, aminoglycoside, tetracycline, macrolides. The RNA synthesis inhibitors are there, and some isins are there. Then inhibition of DNA-dependent RNA polymerase rifampicin are there. That these are mainly work not only as antibacterial, but they are also work as antitubercular. And the last class that I have studied into that I have explained you into that is PC to ATM line Q. Q stands for the quinolones, and they are having ability to inhibit the DNA gyrase. These are also called as topoisomers. So that's all about the classification of antibiotics in terms of chemical as well as mode of action.